Welcome, everybody. We're at ASGCT 22 in Washington, D.C. I'm Greg Slabodkin, Editor-in-Chief of ScienceBoard.net, and I have the privilege of being here with Dr. Bernhard Gentner with Genenta Science. You're a co-founder of the company.
Yes, absolutely.
Just as a quick jump-off point here, just give us a quick elevator speech on the company, how long it's been in existence.
Right, so the company has been founded based on the science and the clinical results that we have developed at the San Raffaele Teleton Institute for Gene Therapy. So, TGET has been working on hematopoietic stem cell gene therapy for decades long. So we have brought several drugs to the market, actually. So and we evolved the technology, from retroviruses to lentiviruses, and learned, I think, how to make this process of ex vivo manipulation more efficient and safer. So the idea that me and Luigi had is how can we exploit this platform for non-hematologic diseases, like cancer, for example. So the whole idea was built on the experience we had in rare diseases, and also on IP that we developed in the lab about regulating transgene expression, because this is critical. Because if you want to exploit hematopoietic stem cells or blood cells as drugs, you want to make sure that you don't have toxicity when you deliver a drug. So you want to have specific delivery inside a tumor, and you don't want to have toxicity in normal blood cells. So what was critical was the combination of a promoter, which is relatively specific for tumor-infiltrating myeloid cells, and microRNAs, exploiting microRNAs to make this expression profile more specific. And so this combination allowed us really to propose a hematopoietic stem cell-based treatment for cancer. And that's how we founded Genenta as the company to make a proof of concept and develop this treatment further.
Interesting. So with the stem cell therapy, you're actually activating the immune system, right, in the tumor. Yeah. So talk to me about how that works.
So the tumor, we have, I think, in the tumor microenvironment, we have two protagonists. We have, on one hand, we have the T cells. They are, of course, dysfunctional, so they're not attacking the tumor because they're immunosuppressive. There are many different mechanisms that make these T cells not work properly. Right. But the second component, which is critical, are myeloid cells. So macrophages, myeloid-derived suppressor cells. So they're a principal component of all tumors, I would argue, all solid tumors. And it's really the interaction between the T cells and these myeloid cells which makes immunity ineffective within the tumor. So we know how to get into the tumor with how to use T cell therapies, Mm-hmm. But this doesn't work alone. In many solid cancers, it's not that effective. And probably the main reason is because you still have this big myeloid infiltrate within the tumor, which continues doing their job in immunosuppressing the microenvironment. So our idea was to get into the tumor by a second pathway through the myeloid cells. So for the T cells, there are stem cell states within T cells. So you can do T cell therapies working with mature T cells because they can propagate and they can expand when you transplant them. We don't really have a macrophage stem cell,
Right.
So in order to get myeloid cell expansion and turnover, you need to go to the true hematopoietic stem cell.
Right.
So because if you were working, let's say, on macrophages, they wouldn't be, I would argue, they wouldn't have a long duration, long durability. They would just persist very shortly and wouldn't be able to expand.
Right.
So that's why we went for the true stem cell, in order to have really an in vivo expansion of your genetically engineered cells and have a durable source. Because once you engraft them in the bone marrow, they contribute for prolonged periods of time to myeloid progeny that are recruited into the tumor. So,
wow. It's interesting. I mean, your work on solid tumors, everybody talks about, CAR T therapy, CAR T cell therapy, and, how much has been done in terms of breakthroughs with blood cancers. But solid tumors are really the holy grail, right?
Absolutely.
Absolutely. And so can you talk a little bit about the challenges, though, of solid tumors as opposed to, blood cancers?
Of course. So first of all, I think we don't have that good antigens, which are tumor-specific in solid cancers compared to blood cancer. That's, I think, the one big reason. In blood cancers, we have lineage-specific antigens like CD19, which are very highly expressed and which are highly specific for B-cell lineage. So the T cells, they work very well against these highly expressed antigens. On the other hand, we also have, if we deplete B cells, that's not going to be a problem, at least temporarily, because you can substitute immunoglobulins. So you can live without B cells for some time.
Sure.
In a solid cancer, things are a bit different because we don't have these good antigens, and we don't really have this delta between the normal tissue and between the tumor to fully explore. That's one reason. The second reason is the myelosuppressive microenvironment, as I said before. So the tumor is much more difficult to access compared to a blood cancer.
Right. And here at ASGCT, you have presented some data, right? Early data from, I think, phase 1 and 2A, right? Trials.
Yes.
Can you talk a little bit about that?
We have treated so far 16 patients with this strategy. This was a phase 1 trial, so we had to evaluate safety, of course, if this approach is safe and feasible.
Right.
And we did this in glioblastoma patients, which is really a terrible tumor which has no cure today. So, especially the subgroup of GBM patients that we treat, so those that have an unmethylated MGMT promoter, so they have a median survival of 12 months, 12 to 14 months, so really very, very short, and no treatment available. So we performed a dose escalation study in these patients. After diagnosis, after surgical debulking of the tumor, the patients undergo autologous stem cell harvest. We produce our product, so we insert into their stem cells a cytokine, which is thought to unleash, to override this immunosuppressive state within the tumor microenvironment. The patient then, while we release the product, the patient receives radiotherapy, which is part of the standard of care. Yep. Then the patient is going to be infused, receives some chemotherapy conditioning to be able to engraft the stem cells, and then receives the product. And we did this in a way that we combined the product, the genetically engineered product, with supporter cells to make sure that we don't see toxicity. So we started with low dose, 0.5 million cells per kilo, and now we're currently dosing at 3 million per kilo because we did not really observe dose-limiting toxicity by that. So we got a very specific release of interferon alpha, that's our cytokine, within the tumor, and very little systemic release of interferon alpha. And I think that's really important because these cytokines, we know they're effective, and interferon alpha has been used as an anti-cancer drug for decades. But the problem is it's not tolerable. It's really not tolerable. So now with this treatment, we get rid of the systemic side effects of this treatment, while we maintain or even get a better local dosage within the tumor, without having also these peaks and troughs that you would get with systemic dosing. So we have a continuous release of this interferon within the tumor microenvironment. And we did some molecular analysis by single-cell RNA sequencing where we can now really say we do see reprogramming of this microenvironment. So the macrophage compartment changes shape. So from being, let's say, an M2-like microenvironment with pro-angiogenic functions, it becomes more an inflammatory microenvironment. So activating in some patients some T-cell responses. And I think that's very promising. That's a very good starting point with which actually encourages us to go further and to.
It's really interesting. I mean, the whole idea of gene therapy and its limitations, right? So often delivery is what is focused on. You know, what are you doing in those areas to help address those challenges?
So at the moment, it's based on hematopoietic stem cells. So the delivery is that you have to engraft these stem cells in the bone marrow. Right. Once you have succeeded in doing that, the delivery is automatic because the stem cell potentially for decades, for years, for decades, produces myeloid progenitors which are called into the tumor. So it's an endogenous delivery of the tumor infiltrating macrophages, if you wish so. I think there are ways how you can further improve the process of getting your stem cell graft into the patient. You know, because up to now, we need conditioning. So we need chemotherapy to successfully engraft these genetically engineered stem cells. Right. And this is, I think, a future development for our technology, you know, to reduce these cytotoxic agents and to be able to engraft genetically engineered stem cells without creating, let's say, side effects or immune suppression from these chemotherapy agents. And I think there's some interesting development going on, like specific antibodies that target stem cells in the body without causing immunodepression. There may be some local strategies to engraft your stem cells in the body. In the future, there may be in vivo delivery. That's what we hear in the plenary sessions. That's what people dream about. You can just inject the vector. The vector will find its way into the stem cells without you having to take them out and do the ex vivo process. So I think that would be something very, very interesting for the future to make also this treatment more sustainable. We still need a lot of work to do to get there.
Yeah.
So if I say one advantage for this cancer gene therapy approach is that we don't need a full chimerism. So preclinical modeling tells us that it should be enough to have something like between 10 and 20% of genetically engineered cells to get a full therapeutic efficacy. So the bar is a little bit lower, like for sickle cell disease or for genetic diseases where you need to get 50, 60, 70% of your stem cells modified. So that could be an interesting testing ground, let's say, for new conditioning methods or in vivo gene therapy.
You mentioned autologous and the whole debate of autologous versus allogeneic. I mean, so what are the pros and cons of both? And are we eventually going to get to allogeneic?
Yeah. So I think that the huge advantage of autologous cells is that you don't have to overcome immune barriers. Right? You don't have graft versus host disease. You don't need immunosuppression. And so I think this is for sure a preferable way to go. There are challenges, of course, in the costs of manufacturing these autologous products. That's a huge challenge, let's say, for the for the current ex vivo process. If one will be able to do in vivo gene therapy, you will not have this problem anymore. No. So let's say when you go for hematopoietic stem cells, I think allotransplantation is being performed for specific hematologic malignancies. And that's where an allogeneic product could be thought of, but only, I think, for these indications, for hematologic indications, where allogeneic transplantation is state of the art at the moment. So I don't see that we can implement allogeneic stem cell transplantation for solid cancers, because that would cause lots of complications. So we would go for an autologous product here.
Very good, very good. Well, Dr. Gentner, it's been a pleasure talking with you, and we look forward to watching future developments from your company. And this is another video from ASGCT, and stay tuned for more from the conference here in Washington.